Hi, my name is Liam Revel, and today I'm going to talk about a new phylogenetic method in which we fit a variable rate Brownian evolution model to quantitative trait data in a phylogeny using penalized likelihood. Typically, when we study quantitative trait evolution on a phylogeny, we fit one of a handful of different models. In the simplest of these, we just assume a constant process across all the edges of the tree. A number of other models and analytical approaches have been developed to allow the rate to vary from edge to edge. For instance, these include the set of methods where researchers fix the mappings of two or more regimes onto the clades or branches, and then fit a model in which the parameters of the process are allowed to differ between these different regimes. This type of analysis is well encapsulated by an article by O'Meara et al., published in 2006, and by the R package AUI by Beaulieu and O'Meara, which extends this to a multi-regime OU. Alternatively, we can also let our data for the trait tell us when or where on the tree the rate or process of evolution might have shifted, or even if it changes as a continuous function of time or of diversity or in distinct periods. This type of approach has been described and implemented in a range of papers and software. Invariably, a key feature of all of these different models is that they require that we compute the probability of our data under the model. This is the quantity that we normally use if we wanted to identify the parameter values of our model that maximize the probability of obtaining the data that we've got, in other words, the maximum likelihood estimates. To get this probability, we need to first figure out the among species variance-covariance structure that's implied by the relationships and branch lengths of our tree and the parameters of our evolutionary model. This covariance structure corresponds to an n by n matrix for n tips, and under a simple Brownian motion model, say, it would be composed by the heights above the root of the most recent common ancestor of each pair of species multiplied by the single rate of evolution under a model, sigma squared. Here, I've imagined a single value of sigma squared 2, and then we can easily proceed to compute the among species variance covariance matrix for the species in this 5-taxon phylogeny by simply taking the product of the length of each edge of the tree and the evolutionary rate on that edge, and then summing these products up the tree to the common ancestor of each pair of species. Now, a colleague recently asked me if there was a simple way to relax this assumption of a constant process or a process specified a priori by the user and ask how the data indicated that the Brownian rate of evolution varied across the edges of the phylogeny. Now, I should say that there are already methods that allow us to fit a model where the data is permitted to inform us about the locations of discrete evolutionary rate shifts on the phylogeny. For instance, the Bayou package by Joseph Wade and other related work. But what I wanted to do was go in a slightly different direction and allow the model to be fully flexible, in which the rate changed, say, from branch to branch on the phylogeny, perhaps under some evolutionary model like geometric Brownian motion, rather than in discrete locations or at specific time points. There's already been some work done on this as well by, for instance, Chris Venditti using Bayesian MCMC, and I was curious to see what we could do using likelihood. In this case, the first step is to write down the probability density of our trait, given a rate parameter sigma squared that varies across the edges of the tree. This is actually pretty easy because we do it exactly the same way as for a constant rate of evolution of the trait. We just take our branch-specific Brownian rates, multiply each of them by the edges in the phylogeny, and then accumulate these products up the branches of the tree. Looking at this problem naively, you might think that we could find the maximum likelihood estimates of the evolutionary rates across all the edges of the phylogeny by just taking the probability density of the data under our model, remember our variable rate Brownian evolution model, and then multiplying this quantity by the probability density of our rates under a model of geometric evolution of sigma squared. Unfortunately, this just doesn't work. I'm not going to elaborate too much on why, but suffices to say this expression will always maximize with zero variation in the rate among edges, regardless of how variable the rate of evolution actually is. The solution that I propose to use is what is called penalized likelihood or regularization. In this case, we take the probability density of our data under the model given our set of sigma squared values among the edges of the phylogeny. And this is the same probability density as before, but I've written it here on a logarithmic scale. And then we multiply this quantity by the probability density of our rates under geometric Brownian evolution of sigma squared. This is our penalty term. And our penalty term is finally multiplied by a penalty or smoothing parameter, lambda. The reason that it's appropriate to call lambda a smoothing parameter is because this function should allow lots of variation between edges if the smoothing parameter value lambda is low. There's then very little penalty for wide variation in rates between branches or different parts of the phylogeny. Conversely, when lambda is large, the penalized function should converge eventually to a single rate Brownian evolution model. To test the method, I simulated a phylogenetic tree and then data for a phenotypic trait under 4 different scenarios for variation in the evolutionary rates among edges of the phylogeny. I then fit our evolutionary model to these data using our penalized likelihood approach and compared the fitted evolutionary rates to their true values. I first simulated rate variation among edges under a model of geometric Brownian motion. This is Brownian motion on a log scale, and it's the model that we're assuming in our penalized likelihood method. This is how data simulated in this way look. Here on the right, I'm showing the phylogenetic tree on the left projected into our trait space. What we can clearly see is that the edges with high simulated rates of evolution, shown here with redder colors, tend to also exhibit larger changes in the phenotypic trait. After simulating these data, I then fit our model using penalized likelihood for various values of our penalty coefficient lambda, and then I compared the edge-specific rates fit under this model to the generated rates in simulation. In each figure panel, I plotted the true rates on the horizontal axis and the estimated rates on the vertical axis. In this case, we see a strong correlation between our generating and estimated sigma square values across all values of lambda. That's good because these data were simulated under our assumed model. In this specific case, the stronger correlation is observed for lambda equals to 1, but this particular best value of lambda will really depend on the scale of our data and the phylogeny. Next, I simulated randomly assigning values of sigma squared to the edges of the phylogeny. Here, I show the phylogeny projected into our trait space again for these simulated rates. When we fit our model to this data, we can both see that the correlation is substantially weaker. Again, this makes a lot of sense because these data were not generated under our assumed model. But we also see very clearly the effect of increasing our value for the penalty term lambda. For higher and higher values of lambda, sigma squared converges across all the edges of the phylogeny towards a single value, just as we'd expect. I also simulated data with discrete shifts in the rate, shown here for a 3-rate model. Once again, redder branches correspond to higher evolutionary rates, something that's also evident in our trait projection. In this case, I once again fit our model using penalized likelihood, but now I compared the estimated evolutionary rates by edge to the mean evolutionary rate across all the edges in a given regime. We can see that in this case, the mean rate, shown by the white dots, quite closely approximates the true evolutionary rate of each of the simulated regimes, especially for intermediate values of lambda, but really across all of the values of the smoothing parameter that I tried. Finally, I simulated under a constant evolutionary rate across all the edges of the phylogeny. In other words, a single rate Brownian motion model. In this case, I compared the mean estimated rate across all edges to the maximum likelihood single rate for a 1-rate model. We can see that the mean is similar to the single rate maximum likelihood estimate for all values of lambda, and the variation among edges decreases with increasing lambda. In addition to this simulation analysis, I also applied the method to some empirical data, actually a very old data set and tree from mammalian body size evolution from Garland et al. 1992. When I do this across 4 different values of lambda, we see some commonalities in the pattern of rate variation among edges across these very smoothing coefficient values, but there's also some important differences as well, which naturally leads to the important question of how do we pick a good value of lambda? One sensible suggestion that has been used for other penalized likelihood analyses, including in phylogenetics, is cross-validation. In cross-validation, we systematically exclude a subset of our observations, refit our model, and then compare the parameter estimates from the subsample data to the estimates from our full data set. I think this would be a sensible thing to do with this model. However, it's really computationally intensive because it requires that we fit our model many tens or hundreds of times. So it's something that I'm still working on. Lastly, anyone who's interested in learning more can already check out a bioRxiv preprint that I've written and posted publicly about this method, or my phytools blog. That's it.